DrBeen.com. Welcome to one more show. So the discussion today is a paper, Italian paper by Dr. Carlo Brogna and his colleagues. I believe they are the same researchers, majority of them, who also had that paper that I discussed a few weeks ago about the toxins that are found in the fecal matter of the patients, which paper was then picked up by someone who was claiming that there is snake venom. So this is the same group of scientists. The discussion that they are doing today, and I think it is very useful. I was not very impressed with the previous one. And there is a clarity in this paper. They talk about the other paper, and they say that those toxins were probably produced by gut microbiome instead of some toxin produced by the virus. That can make sense. So what is the discussion that we're going to do today? Dr. Carlo Brogna and his colleagues, they did immunofluorescence and electron microscopy of the bacteria that are present in our gut. So they collected the stool samples from SARS-CoV-2 infected patients and healthy controls. And what they wanted to see was if there is SARS-CoV-2 proteins, if these proteins are present inside the bacteria. And they proved, at least from their study here, that they saw SARS-CoV-2 present in the bacteria. That means SARS-CoV-2 has a potential to disrupt our microbiome for the gut. Microbiome are the microorganisms, microbes, bacteria mostly, that are present in various parts of our body, and some of them are helpful and some of them are harmful, and usually there is a balance between them. So when something comes in, for example, SARS-CoV-2, and it disrupts that balance by infecting the bacteria, or maybe by disrupting the good bacteria, then we can have severer outcome of the disease or unwanted outcomes. So let's look at this paper. It's a very interesting paper. So here is the PDF. Could SARS-CoV-2 have bacteriophage behavior or induce the activity of other bacteriophages? Bacteriophage is usually a virus that can infect a bacteria. And then there are multiple life cycles. There are multiple lysogenic or lytic. We'll talk about that. But a virus that can infect a bacteria. So let's start our discussion. I have done some drawings here. Let me quickly show you the references. So this is DrBeen.com. In the description, there is a link to DrBeen.com. Super affordable price plan, 1,000 videos, less than $100 price. Check it out. Here, this is the MDPI. This is about MDPI. It's a pioneer in scholarly open access publishing. This is the paper itself. Then here is the bacteriophages and their life cycles. We'll discuss them. This is just reference. This is polio vaccine. And why do I have it over here? The authors suggest that it may be interesting to create oral weakened virus vaccine, just like Sabin formulated oral polio vaccine, because that vaccine will be needed for the gut bacteria. Then this is an article in Mayo Clinic about what are prebiotics versus probiotics and what other things. I think at the end of this whole discussion, the takeaway is going to be, what can we do? And here is, I think we should just start from here. So we should acknowledge that the study says in our gut bacteria, the SARS-CoV-2 can infect them and disrupt them. That means our gut health can become bad, one. Secondly, we can have a severer outcome because the gut health is bad. Thirdly, other diseases, other autoimmune issues, other immune dysregulations could pop up because SARS-CoV-2 caused a problem with the gut bacteria. So we should just acknowledge that that can happen. Then one simple at-home remedy, one can take probiotics or prebiotics. Probiotics examples are yogurt. They have the organisms, gut microbes, and sauerkraut. Sauerkraut is here. Sauerkraut is actually, I believe these are cabbages. These are thinly sliced and they have organism on this. So it's finely cut raw cabbage that has fermented by various lactic acid bacteria. So it has bacteria on that. So uh, sauerkraut, yogurt are probiotics. They have the pathogens in them, just like Arby's have the meat, right? And the prebiotics, these are the foods that can nurture our friendly bacteria. These can be the food for the bacteria. So, for example, whole grains, bananas, greens, onions, garlic, soybeans, and artichokes. When you take them, you are actually giving food to the bacteria that are sitting in the gut, and that can help them grow. So I thought we'll take the takeaway first. Now let's look at my diagrams. So here we have a little poopy looking environment. That is because we're going to talk about the gut microbiome and fecal matter. So this study is an in vitro study, small sample size, 3 patients who were SARS-CoV-2 positive, and they had their fecal samples or the gut microbe samples for another study, and they used them for here as well. Similarly, they had 3 healthy patients as well. I think a total 9 samples they had. So they had samples from the uninfected patients and samples from the infected patients. I shouldn't say uninfected patient, uninfected people. The question in their mind was the infected people, patients, their gut bacteria, will they have SARS-CoV-2 in them? This, according to the authors, is the first study of this type. And this is the first time they are proving yet that this is possible. This is the first ever study. And authors kind of were surprised a little bit that it is 2.5 years or more than 2 years that we have been, all of us, doggedly discussing if the SARS-CoV-2 enter a human cell, or we call them eukaryotic cells. Eukaryotic means true nucleated kind of cells. So the cells that we have, or mammalian cells. So they said we are researching how do these viruses enter the cells. We know about ACE2 and we know about CD147 and all that. But they said there is very less research on if this virus enters the bacteria in our gut and what will it do. So this is their first study. So the summary of the study is they can actually prove that SARS-CoV-2 can infect the bacteria of our gut. So that is the new information they're providing. What they're showing is, now the question should be in your mind right now, and that is, how does SARS-CoV-2 infect a bacteria? Does the bacteria have an ACE2 on it, or CD147 on it, or some other receptors on it that the SARS-CoV-2 uses to enter the pathogen? And that is where the answer is no, it has a different mechanism. So let's look at these two quickly. We know that for mammalian cells, meaning our cells, for example, or other animals, the SARS-CoV-2 uses ACE2. And now we know also CD147 and some other mechanisms too. For the bacteria, it is very interesting. They suspect that what happens is when the SARS-CoV-2 arrives near the bacteria, so imagine near the end part of our gut, where there is fecal matter and where there are lots of bacteria as well. Imagine their SARS-CoV-2 arrives there too. And these bacteria look at SARS-CoV-2 and they start releasing toxins to try to destroy this virus. And virus enters. So what happens is virus's surface changes. Why? This is just like if you take meat and you put that in a cooker and you add some acids to it or some spices to it, that meat is going to start changing its shape, or its surface is going to start getting changed. Same way, when SARS-CoV-2 is present, in the presence of these toxins, the toxins would cause the surface of the SARS-CoV-2 to change. It will become rugged and rough. It will become modified. Now that modified surface virus interacts with the bacteria and is able to enter those bacteria because of those modifications that bacteria made trying to protect themselves. So that's beautiful. In addition, I mean, beautiful in a bad way, right? I'm a mechanisms guy, so I like these mechanisms. The other thing is that these bacteria, inside them, they have proteolytic enzymes. Almost all cells have proteolytic enzymes, or the enzymes that can break down proteins. So when these bacteria sense the pathogen near them, they release the toxins, plus they release the proteolytic enzymes. They all change the surface of the SARS-CoV-2. That enables the SARS-CoV-2 to be able to stick to the pathogen, the bacteria, and enter it. So this possible mechanism is also a unique mechanism for SARS-CoV-2, because the other viruses that can enter a bacteria, they have specific receptors. This virus is something. Okay, so continuing. Now, once the bacteria are infected, and they saw this in their cultures, they saw some bacteria. So there are hundreds and thousands of kinds of species that are living in us. Some are good, some are bad. They saw that some bacteria started dying. Some bacteria started becoming, their count started reducing. And some other bacteria started growing. And this is actually a normal process in our body, in our gut especially, that when one kind of normal flora, or let me back up for a second. Imagine our gut is a universe. In that universe, there are multiple countries, and every country has a different bacterial species that lives in there. When one species gets destroyed by some mechanism, for example, here we are seeing SARS-CoV-2, other viruses do it as well. Sometimes we take antibiotics and they can disrupt that as well. Sometimes immune system can cause dysregulations. So imagine one country with one species got destroyed. So gut doesn't do this to say, all right, this patch of gut would stay empty now. There will be no bacteria here because this country belonged to bacteria, let's say Cereus, Bacillus cereus. It should not be that. That's a dangerous one. But anyways, let's use an example. And now that they are not here, we'll just wait for them to come back. So we'll leave this country alone. That doesn't happen. In fact, the other bacteria will then start growing more and occupy that place. So they said that in the culture, they saw some bacteria whose count started going down and some bacteria whose count started going up. It is not that SARS-CoV would kill every bacteria there. It would kill some and the others would then overgrow. That is the dysregulation of the microbiome. That is what's going to cause a problem with the immune system as well. That's what's going to cause a problem with the digestion as well. That's going to cause a general health issue. And they say that that would then be connected to the severity of COVID as well. So this is another point that they observed. Then I just talked about it. Severity of the COVID will then also be impacted. Then they say that this discovery, and I would show you the diagrams afterwards, this discovery that the virus is present inside the bacteria is infecting it also has a diagnostic value. That means we could actually do fecal samples and then see if the SARS-CoV-2 is present, and then see what is the intensity of the presence and so on. So there is a diagnostic value as well. Then they say there is a epidemiologic value as well. Why? Because if the pathogens, the bacteria of the gut have the virus, these bacteria are infected, then these bacteria are released in with fecal matter in the wastewater. Then not only just the wastewater, fecal oral transmission is a process as well, where somebody's fecal matter has some infection in it, and they are, let's say, not very hygienic, and their fecal matter is touched, and then that they touch other surfaces or food material and so on, and now others will catch that, who would touch that and touch their mouth. This is fecal orophecal route or fecal oral route. So they said we should seriously think about SARS-CoV-2 being fecal oral transmitter as well. And they say that we are aware that we see these SARS-CoV-2 in wastewater. Important thing that we are missing is to actually look at the bacteria as well, to say, is it present in the bacteria? How much? And what is the situation of the gut for each person? Okay. Then they say there is a therapeutic value. Therapeutic value is that, number one, just like I said, here are some prebiotics and probiotics. Number one, you can look at how do we keep gut microbiome health. Number two, there can be vaccines that are oral vaccines, which are made for bacteria, not for us. That is, you take attenuated virus, you give the oral drops, and imagine that that attenuated or weakened virus is actually going to be picked up by the bacteria, and that would allow bacteria to figure out how to handle this virus. It's more of a vaccine for bacteria so that bacteria health can be sustained. So they say over here, I've taken an excerpt from their document. It must be also remembered that bacteria are found on oral, nasal, pharyngeal, alveolar, and intestinal mucous membranes. Oral, mouth; pharyngeal, the back of the mouth or the throat; alveolar, the lung, where the gas exchange is occurring; intestinal, gut. Mucous membrane are the surface membranes, the wet surfaces, and are often more numerous than epithelial cells. And they say that the bacteria present on these surfaces are actually more than the cells of our surfaces. Even if a virus does not replicate in them, the surfaces, the cells, and the bacteria, this does not imply that it will not cause discomfort through the bacterial flora of the human being. So even if the virus does not infect a cell, it can still infect the bacteria, and that would cause the normal flora. Normal flora means the normal bacteria present on us. It would disrupt them, and that would cause disruption. Understanding that a virus also binds to, interacts with, and infects bacteria, and that fecal oral transmission is an additional source of spread, completely changes the epidemiological scenarios of SARS-CoV-2. And check this out. It changes how to intervene. So how do we manage SARS-CoV-2? How to give policy input. So what are the policies? How to treat COVID-19 patients. For example, fecal oral transmission has a different set of hygiene requirements. COVID-19 patients, and at the onset of the symptoms, what management to do, and how to prevent, and perhaps another possible solution could be the vaccine, as Sabine said. So this is the Sabine vaccine, or the oral poliovirus. So the Dr. Sabine, who invented that vaccine, said an attenuated virus for oral administration, probably justified by the need of the bacteria of the microbiota to interact with the virus. Therapeutic value, epidemiological value. Now, they also say in their paper that this proves, and I'll show you the diagrams in a second. They say that our research proves that the SARS-CoV-2 and the bacterial interaction is both lytic and lysogenic. And I know that healthcare workers and professionals and students, healthcare, they know this. I just want to very quickly explain that here as well. So it's a quick explanation of what this is. Imagine this guy is a bacteria. Bacteria have viruses that infect them. Those viruses are called bacteriophages. So here is a bacteriophage that is connected with the bacteria with some receptor, and it's going to send its material DNA inside the bacteria. So if you see here, this red part is the virus's genome or genetic material. This little black loop is the bacteria's own DNA. In the lytic phase, lytic means lysis, lysis means breakdown. In the breakdown type of cycle, what happens is once this piece of DNA comes in, or the viral genomic material comes in, that material would start making more viruses. And so that genetic material will give rise to other proteins, and those proteins would start helping make more viruses, just like it happens in our cells too. In that process of making viruses, the bacterial DNA is broken down. So that is like bacteria's brain is crushed and used up to make more viruses as a raw material. And not only bacterial DNA is taken up, majority of the bacterial proteins and things would be taken up as well. The only thing that would be kept is the machines inside the bacteria that would help make more viruses. The end result is this bacteria is going to break down. Of course, it's going to break down. Everything is getting destroyed inside. So when the bacteria breaks down, the newly formed viruses will come out. This is called lysis of the bacteria, and this phase or this cycle is called lytic cycle. On the other hand, this is also possible that the bacteriophage infects the bacteria. Then instead of actually destroying the bacteria right away, the genetic material of the bacteriophage becomes integrated with the genetic material of the bacteria. And now, whenever the bacteria divides, that genetic material divides as well. So now this virus's genetic material is in a dormant way, in a silent way, getting increased in number. At some point, under stress, this piece of the viral genetic material will become separated. Sometimes these are called plasmids, and bacteria actually use them as a superpower. So these plasmids are separated, and if it is a dangerous virus, then this lytic cycle would start. This cycle, where the virus just becomes part of the DNA, that is called lysogenic cycle. The lytic cycle can convert into lysogenic. Lysogenic cycle can convert back towards lytic as well. This is how virus can live with the bacteria. They say that SARS-CoV-2 could be doing the same thing inside our gut to our bacteria. Their conclusion is, and there are a couple of things that are interesting here. One is this: SARS-CoV-2 has two mechanisms of actions. So this is they are the first one to say two type of infections: our body cell infections and our bacteria's infection. Bacteria itself is a plural. So two mechanisms of action. It infects both the eukaryotic cells. The eukaryotic are true nuclear cells, that is our cells, as reported in the current literature, which they say everybody is talking about that already. But it also infects the human bacterial flora. And they say two bacteria are already identified. So in their own studies, they have actually identified two bacteria. They said data is in preparation. I'm sure they're going to write another study. So then they say, in this case, we could consider the virus as a bacteriophage with a lytic phase and a lysogenic phase. Good job. Now, let's look at their diagrams, which the meat of the whole thing is the diagram. So let's look at it. These are beautiful diagrams, and I hope they're okay if I show you those diagrams. These are present on their paper and in their PDF. So first here, The only thing is diagrams quality is not the best. So if you can see, you cannot really read very well. But these are uh, various donors. And then within the donors, the growth of the SARS-CoV-2 within the bacteria. So this is that graph. What this is showing is the bacteria, the SARS-CoV-2 grows in the bacteria. That's one. This is more interesting, these diagrams. Here, this green one says gram positive. So in a stool sample, and they did a lots of culture and the methods were beautiful. So they did the culture and they identified these areas where there are bacteria. Then what they did was, remember that SARS-CoV-2 has nucleocapsid proteins. So they had antibodies against the nucleocapsid, right? So if they put the antibodies on this thing, and if the virus is inside the bacteria, then the bacteria where it is, that bacteria would show nucleocapsids in it. And if you see on the right side here, nucleocapsid protein, and they are showing that these are found almost at the same place where the bacteria are found. So if you overlay them here, this is the merged diagram of both of them, then wherever the bacteria is, that is where the nucleocapsid protein is. That means that nucleocapsid protein is actually inside the bacteria. And if you don't agree with that, I'll show you more diagrams that they have of electron microscopy where they actually show the pathogen, the virus in the bacteria. And so if you see here on the right side, this is the gram positive bacteria, this is the nucleocapsid protein, and then they are overlaid. And if it is a control, that is a person who is not COVID positive, and their fecal matter and poop is seen, then there is no nucleocapsid seen. Beautiful work. I was not very pleased with their toxins work. It was actually used by incorrectly, but anyways, this is a good work. So check this out. Look at this. This one here, these are bacteria, these big blobby things, and these red arrows are showing where the bacteria is breaking down. This is lysis. Remember the lytic cycle? These black things are the antibodies connected with the SARS-CoV-2 antigens. So these are the viruses. Viruses, SARS-CoV-2. This was not known that SARS-CoV-2 is actually a bacteriophage. So here it is inside the bacteria. It is increasing in number. And similarly, if you see on the right side, these are SARS-CoV-2. And why they have put the golden arrows there, they actually used the test with antibodies that was called gold test. So that is why the golden arrows. So here, so what they're showing in these two diagrams is number one, bacteria are being broken down. That is lysis. And number two, viruses are in them. That is bacteriophage behavior of SARS-CoV-2. Then again, if you see here, a similar thing in this diagram F, you are seeing some parts of the membrane that are breaking down, and then you're seeing some black dots, that is the virus. Here again on this is G. If you see these red areas are the bacteria breaking down, red arrows, and then the golden ones are the ones that where the pathogen is. And I believe this one here, this, if you read here carefully, it says H on it. So the H is actually control. So if you see the control, their bacteria are healthy, their bacterial walls are fine, there are no SARS-CoV-2 or the nucleocapsid antibodies seen in them. So this is a healthy gut bacteria culture. This is unhealthy. So whoever had them, their gut microbiome for sure got disrupted. So these are the diagrams. And what they did was, in their culture, they made sure that one could say that, hey, this is really not the bacteria. Any cell could have the pathogen in them, and we are just seeing those cells. So what they did was, in their test, in their lab, They cultured the bacteria, and whatever was the fecal matter would have cells and bacteria both. They cultured that for 30 days. In that 30 days, in that culture medium, during that 30 days, the mammalian cells or eukaryotic cells die. They do not have enough nutrition for those cells, so they just die. And and excuse me. And by the end of 30 days, what is left are bacteria. They only give the nutritional substances that are good for bacteria. They do not give the substances for the cells. So the cells die out, bacteria are left. Then they can study those bacteria. That is what they had done. Beautiful study. And now, slight changes to my, what is this? My studio. If you see here, my degrees. So there are 2 or 3 more degrees. I didn't have space for them. So these are really my medical and engineering degrees here. So I put them here, mic here. My other camera broke down. So I sent it out for repair. And this is a different lens. So you can actually see it is a little wide angle. And that is what we have. Please do me a favor, like, subscribe, and share. That is the easiest thing. I hope you would like this kind of work to continue. I was thinking over this weekend, in the whole YouTube landscape for medical teaching, there is actually no one who's teaching mechanisms. There are people who are doing reporting. US has this, UK has this, South Africa has that. Today we have surged, tomorrow we have not surged, and so on. There are folks who would do once in a while some technical discussion, but a daily discussion of these mechanisms is not there. And I think not just COVID, we should continue this for others too. If you would like to support this work, there are links in the description. Easiest is to buy DrBeen's premium account. It's really, really affordable. Check out that link. And it is one-time payment for lifetime. And there are 1,000 videos there. In addition to that, there are links for Buy Me a Coffee or PayPal or becoming a Substack member or becoming a patron. Thank you very much. I prefer Substack or patron type memberships. And I would see you on the Cool Bean Cafe side in a few minutes. And those who may not know where the Cool Beans Cafe is, you can actually just search for it, Cool Beans Cafe. I'm going to give you the link here as well. It's called Cool Beans Cafe Live. And here comes the link. Alexander says, I'll buy DrBeen account. All right. So thank you very much for the super chats. There are lots of them. Thank you. And I will see you on the Cool Bean side, cafe side in a minute. I hope there was no question and I didn't miss those questions. We can do the questions about this talk over there as well. So see you there.